I think this is now. Yes, good morning, dear students. আজকে যে ক্লাসটা স্টার্ট করবো আমরা সেটা হচ্ছে যে the next part of the structural organizations in animals. Okay? So আগের দিন আমরা কি পড়েছিলাম? আগের দিন আমরা পড়েছিলাম হচ্ছে epithelial tissues. So epithelial tissues we have already covered. Even on the last day we have talked about glands, different types of glands. And রুকো এই প্রশ্ন দেখতে পাচ্ছি না. Yes. We have just started off with the connective tissue. Fine. So we have started off with the connective tissue. Though it's not completely over, so we are going to start from blood today, I think. Yes, blood. Bone is over. Bone is not over. So we are going to start from blood. So let us start with the blood portion. Okay. Blood portion. Now see, when we talk about blood, the study of blood is called hematology. The study of blood is called hematology. Fine. Now when we talk about blood, blood is what? Blood is a fluid connective tissue. Blood is a fluid connective tissue. It's a fluid connective tissue. Now, why it is called fluid? Because the gap between two cells are huge. Consider if this is a blood vessel. If this is a blood vessel, in between the two blood vessels, you can see RBC is present. So this is RBC, this is another RBC. Now, in between two RBC, you can see what is present is intercellular space. What is present is intercellular space. Fine. Intercellular space. And this intercellular space is huge between two RBC. This is one RBC, this is another RBC. So in between two RBC, you can see there's a huge intercellular space. The more the intercellular space, the more is the chances of it to become fluid in nature. Any issues? So blood is a fluid connective tissue. Blood is a fluid connective tissue. The second point about blood is what? The pH of blood, it is slightly alkaline in nature. It is slightly alkaline in nature. Alkaline in nature. And what is the pH? The pH of the blood is 7.34. The pH of blood is 7.34. It's a slightly alkaline in nature, 7.34. Okay? Now, when we talk about blood, we can classify blood into two. One is plasma, which is 55% of the blood is plasma, and the rest is blood corpuscles. The rest is blood corpuscles, which are also called as formed elements. Which are also called as formed elements. Now, blood corpuscles are 45%. So 45% of the blood are blood corpuscles. Now, when we talk about plasma, we can classify plasma into two. One is organic portion and the one is inorganic portion. Just organic portion contains mainly the protein. And what are the proteins? There are four different types of proteins. Basically, they have got albumin. Also, they have got globulin. Prothrombin is the next one. And fibrinogen. Fibrinogen is another type of blood protein, plasma protein. Out of these four, the first one, albumin, is the most abundant protein. It is the most abundant protein in the human body. So if the question is, what is the most abundant protein in the human body? The answer is albumin. So it is the most abundant protein. Okay? Most abundant protein. What we can expect in Neet? Name the protein which is protective in function and present in our blood. Name the protein which is protective in action and helps in what? Protection. And the answer to this question is globulin, which is also called as immunoglobulin. Immunoglobulin, and we know that the antibodies, they are basically all immunoglobulin in nature. We call them as IgG, IgA, IgD, IgM, and IgE. So these are globulin. Prothrombin and fibrinogen, these are the two plasma proteins which helps in blood coagulation. Clear? So this is about organic portion. When we talk about inorganic portion, we can classify inorganic into two. One is anions and cations. Anions and cations. Anions and cations. Anions and cations. Yes. When we talk about anions, they are Cl minus, that is chloride ions, and bicarbonate ions, HCO3 minus, bicarbonate ions. Clear? Cations, when we talk about cations, help me with some. It is Ca plus 2, Na plus, K plus, Mg plus 2. So these are the cations present inside the plasma, inorganic portions which are cations. Any issues about that? So this is the classification of plasma. Blood corpuscles, they are also called as formed elements. Now what are blood corpuscles? We know that there are three different types of blood corpuscles. What are they? First is RBC, the second one is WBC, and the third one is platelets. So when we talk about RBC are also called as erythrocytes. RBC are also called as erythrocytes. WBC are also called as leukocytes, and platelets are called as thrombocytes. Clear? RBC, WBC, and platelets. Clear? Any issues about that? Good. Now, let us start with the first one, which is called RBC. So let us start with RBC. What is RBC? How to name it? They are called red blood corpuscles. Now you may ask me, so why they are not called as cells? Because in some of the books, what is being said about RBC, they are called as red blood cells. But mind you, they are, to be specific, they are not cells, they are corpuscles. Now what is the difference between corpuscles and cells? Now normally we know when we talk about cell, the cell should have one particular basic property, that they should show cell division. But corpuscles, they never show cell division. They are formed whenever they are needed in our body. Even if there is a requirement of RBC, the RBC needs to be produced inside the red bone marrow. RBC needs to be produced inside the red bone marrow. Got the point? So this is what? This is red bone marrow. So whenever there is a need of formation of RBC, it has to be produced where? Inside the red bone marrow. And that's why they are called formed elements. That's why these blood corpuscles are also called as formed elements. Okay? Now, so first point about RBC. What is the origin? Where is the site where they are being produced? They are produced inside red bone marrow, and they are present at the two extremities of the bone. Next point. What is their initial cell? Initial cell. They are also called as stem cell. And the initial cell or stem cell is hemocytoblast cell. Hemocytoblast cell. Hemocytoblast cells, they are having a well-defined nucleus. They have got a well-defined nucleus. And normally, what do we know about RBC? That RBC are enucleated in a matured state. That means there is something which is unique in them. That means in immature state, they have got nucleus. They have got nucleus. Now, let us talk about this cell. This cell is having a well-defined nucleus. So, this is a nucleated cell. It's a nucleated cell. When we talk about process of formation of RBC, the condition is called erythropoiesis. The process is called as erythropoiesis. So, what is the meaning of the term erythro? Erythro means red. Erythro means what? Erythrocytes, which has come from RBC. Poiesis means what? Formation. Poiesis means formation. So the process of formation of RBC is called erythropoiesis. Now, this erythropoiesis takes place where? We already know about it. It takes place where? Inside red bone marrow. It takes place where? Inside red bone marrow. Now, the fact about this RBC is the cells, this is a hemocytoblast cell. It's a hemocytoblast cell. And this cell grows in size, and the nucleus, the size of the nucleus gets a little bit degraded, or it diminishes in size. This cell stage is called pronormoblast cell. Pronormoblast cell, or also called as proerythroblast cell. Proerythroblast cell. Okay? Now, this cell size further increases, the nucleus size further diminishes in size, and for the first time, the structure which comes in is hemoglobin. This stage is a very, very important stage, which we call it as intermediate normoblast cell. Intermediate normoblast cell. This cell further grows. And the nucleus is just like a dot now. The hemoglobin, they have further taken huge part of the cytoplasm of the RBC. So the hemoglobin amount increases in the RBC, and the nucleus size, this is the size of the nucleus now. Okay, and this cell is called late normoblast cell. Late normoblast cell. Otherwise, also it is also called as late erythroblast cell. Erythroblast cell. Okay, and then the cell size further increases, nucleus completely gets disappeared, and the whole part of this particular cell is not having nucleus, it's completely covered with hemoglobin. And this cell is now called as reticulocyte cell. Reticulocyte cell. This cell further, it shows what? If this is the front view, they are biconcave in shape. From this part of the RBC, you can see it's oval shaped. But if you see it from this side, they are biconcave shaped. And this is the hemoglobin. What is it? It's the hemoglobin. Now, this cell is now called as erythrocyte cell. That is our RBC, which is not having any nucleus. This is erythrocyte, or else called as RBC. So what is the shape of RBC? They are biconcave shaped. They are enucleated. Fine. If you compare the RBC of human with RBC of frog, so what's the difference? The RBC of frog, they are round in shape and they are nucleated. They are having well-defined nucleus. Can you tell me a name of a mammal having nucleated RBC? Camel. Camel is the only mammal having well-defined nucleated RBC, even in their maturity. This is the process of RBC which takes place where? Inside bone marrow. Inside which bone marrow? Red bone marrow. Now, What is the process just now we have talked about? It is erythropoiesis. But for this erythropoiesis, there is a hormone named erythropoietin. There is a hormone named erythropoietin which is produced inside kidney. And this erythropoietin, they help in this erythropoiesis process. So it's a very, very important step again. It's a very important point. Okay? Next point. When we talk about this RBC, as because the RBC, they are biconcave shaped and they have a huge amount of hemoglobin. Again, what is that amount of hemoglobin which is present inside RBC? When we talk about the hemoglobin content, it is normally for human male, it is 14.5 gram approximation, fine, 14.5 gram per 100 milliliter of blood. This is the content of hemoglobin present inside blood. Now, if this content increases, then it is good for the body, but if it decreases, then definitely, normally we know that the hemoglobin, it binds with oxygen to form oxyhemoglobin and helps in the transfer of oxygen from the lungs to the tissues. This hemoglobin also binds with carbon dioxide in order that the body should be, the body wants to get rid of the carbon dioxide. So in that case, the glucose which is being broken down, by the process of respiration. So when we inhale oxygen, when we inhale air, the glucose gets broken down in presence of oxygen to give to carbon dioxide plus water plus 686 kilocalorie of energy goes out, which is equal to 38 ATP. That means one molecule of glucose can fetch us 38 ATP. And what is ATP? We know that it's the energy currency of the cell. In that case, this carbon dioxide needs to be transported out of the body. So this carbon dioxide again binds with hemoglobin to make what? Carbaminohemoglobin, and also it carries the carbon dioxide out from the body. Though there's other means, and that is in the form of bicarbonates. Fine, 70-75% of the carbon dioxide is transported in the form of bicarbonates. Some amount by the form of carbaminohemoglobin, and some of them they remain in the plasma, dissolved in the dissolved state. So hemoglobin, as because hemoglobin is present hugely inside RBC, that means it helps in the transport of oxygen from lungs to tissues, and carbon dioxide from tissues to lungs. Clear? Any issues about that? So this is about basic idea about the, see, when the hemoglobin is present hugely inside the RBC, that means there are some, why nucleus is not there? To pave the way for hemoglobin to be present over there. Along with the nucleus, what else that the RBC are missing? They are missing endoplasmic reticulum, they are missing Golgi body, they are missing ribosomes. And what about the double membrane structure? So what they don't have is they don't have mitochondria, because they don't need any mitochondria, they don't need any energy to travel, because RBC is being pushed by the pumping organ called heart. So there is always a continuous flow of blood inside the body by the help of the pumping organ called heart. Any issues about that? Now, when we talk about the number of RBC in males, why we are talking about in males? That means in females they are different. In females their number varies. In males the number of RBC is 5 to 5.5 million. 5 to 5.5 million per cubic millimeter of blood. This is the amount of RBC inside males, and we know million means lakhs, that means 50 to 55 lakhs. 50 to 55 lakhs per cubic meter of blood. In case of female, it is a little bit less. What is that? 4.5 to 5 million. 4.5 to 5 million per cubic millimeter of blood. Okay, per cubic millimeter of blood. Now, if this number is increased, if the number is increased than this normal levels, the condition that we call it as is erythrocytosis. Erythrocytosis, which is also called as polycythemia. Which is also called as polycythemia. Emia means blood. Poly means many. Cythe over here means RBC. So many RBC inside blood is called polycythemia, or else it is also called as erythrocytosis. Now, when the RBC count is reduced, the condition is called erythrocytopenia. Erythrocytopenia, which is also called as oligocythemia. Also called as oligocythemia. Oligo means fewer. Oligo means fewer. Now, is this clear? Now, in NEET examination, the question which is very common normally being asked is about this. How erythrocytosis occurs? Normally, they are going to take a subject to a hill, and when a person ascends towards a hill, the oxygen content lowers down over there, and the RBC count has to increase, because the requirement of the tissues for the blood is going to be the same. Say the liver or the other organs like gallbladder, as because you are moving towards a holiday, towards a hill, it doesn't mean that liver is going to say, chalo hame kaam nahi karna hai. We are also in a holiday mode. Fine, so liver is going to do the job. So in that case, liver is going to make the same job. So they are going to ask for the same amount of blood, but as because oxygen is less over there, the RBC count has to increase. The condition will be called as erythrocytosis now. And during this erythrocytosis, as because huge number of RBC needs to be produced where? Inside the bone marrow, inside the red bone marrow. So huge amount of blood needs to reach where? Inside towards the bone. Less amount of blood is going to reach towards your brain. So you are going to feel what? Dizziness, nausea, and to some extent you are going to feel vomiting. Though you can also answer the same by the help of the activity of hypothalamus. Because with every 100-meter rise in your levels, ascending towards 100 meter, with every 100 meter you take over, 1 degree Celsius is going to get decreased. So hypothalamus, within a span of 10 minutes or 5 minutes, you have covered 100 meter of the altitude. You have, ascended in that particular region. So within those 5 minutes, your hypothalamus needs to regulate the body temperature, which is tough for the hypothalamus, and that's why we know that we need a little bit of time for acclimatization. We need some time for acclimatization, and that is needed. Okay, and after 2 to 3 hours, you feel it's okay. Why? Because your RBC count has come down, has increased to some levels. Fine? Yeah. So this is what is important. Erythrocytosis takes place when you ascend towards a hill, clear, or mountain. A hill or mountain. We cannot call it as a hill. It's better to talk about mountain. Fine. Because oxygen is decreased when you reach towards a mountain, which is over and above 1,000 meters. Okay. Now, this is a condition which is very important for NEET examination. Okay. And let's talk about the membrane of RBC. The RBC membrane. The membrane of RBC. So this is hemoglobin inside. The membrane of RBC is called as Donan's membrane. The membrane of RBC is called as Donan's membrane. And I think you have heard about in class 12, you have heard about sickle cell anemia. In sickle cell anemia, the RBC shape becomes sickle shaped because there is a gene which is called pleiotropic gene. We know that pleiotropic gene is a gene which can, normally we know that one gene, one character concept. So one gene is responsible for expressing one character. But in pleiotropic gene, one gene can take care of more than one character. It can express more than one character. So in that case, that gene is called pleiotropic gene, and it makes the RBC as sickle shaped. So if the RBC changes from biconcave shape to sickle shaped, the amount of hemoglobin inside also decreases. The amount of hemoglobin inside the RBC also decreases, which ultimately is called as sickle cell anemia. Though we are going to talk about the different types of anemia. We are going to talk about different types of anemia. Okay, now, so the membrane of RBC is called Brown's membrane. Sometimes in the blood vessels, when the RBC travels, sometimes the RBC can make like stacks, they remain like stacks. And this stack-like structure is called as Rowley formation. Rowley, UX is silent. Rowley formation. So we call it as a Rowley formation. Fine. So this is unwanted. Why? Because if they remain as stacks, it's going to stop the blood to move from one part to the other. And if this happens inside your heart or in your brain, that it can prove to be fatal. Any issues about this? So RBC. Now, what else about RBC? Yes. When it is going to form inside the bone marrow, definitely it should have some lifespan. And what is the lifespan of RBC? It is approximately 120 days. It is approximately 120 days. And what happens after that? What happens after that? A term occurs, a stage which is called as hemolysis. Hemolysis. Lysis means what? Lysis means breakdown. Lysis means what? Lysis means breakdown. So breakdown of RBC, hemo means RBC, is called hemolysis. So when RBC breaks down, now name the organ which helps in the breakdown of RBC. I'm giving you options. Is it pancreas, liver, spleen, or bone marrow? It's a liver. So RBC is broken down where? Inside liver. So liver is going to break down RBC, and ultimately RBC is going to change into two bile pigments called bilirubin and biliverdin. Bilirubin and biliverdin. This bilirubin and biliverdin, both of them, you know that both of them are bile pigments. Both of them are bile pigments. Now this bilirubin changes into starcobilinogen. Starcobilinogen. And this starcobilinogen is responsible for the yellow coloration of stool. So the stool color is yellow, which is quite common because the RBC after 120 days it is going to die off, and ultimately it's going to change into bilirubin only. And this bilirubin is a reason for the yellow coloration of the stool. Now sometimes you can see the stool color is red. That means it is containing what? Fresh RBC. That means there has been some ulceration in the lower gut called rectum. So either it has occurred in rectum or colon part, which is close towards the anus. So you can have a fresh blood coming in, or sometimes you can have ulceration in the upper gut, where? Close to the stomach. So there can be peptic ulcer, there can be duodenal ulcer. So in that case, the blood which is coming out inside the organ, on that particular blood, HCl is going to oxidize the iron into black. And that means the color of the stool is going to become black, and the medical condition for the black colored stool is called melena. It's called melena. So it's an abnormality. If the stool color becomes black stool, we call it melena. It's a medical condition. And why this black stool is occurring? Why? Because if this is stomach, and inside this stomach, if there has been some ulceration, blood is coming out from, oozing out from this part, HCl is going to bind with this blood, and make it oxidize and make it turn black. So this black colored stool is called melena, which is abnormality. Let's talk about biliverdin. When we talk about biliverdin, this biliverdin is also a bile pigment. And this biliverdin changes into urobilinogen. Urobilinogen. Uro means what? Urine. Uro means what? Urine. Emia means what? Anemia. Emia means blood. And this uro means what? Urine. So urobilinogen is a pigment, and this ultimately changes into urochrome. Now, what is the meaning of chrome? Chrome means color. Uro means urine. So it provides yellow coloration to urine. It provides yellow coloration to urine. Again, let me tell you, yellow color of urine is a normal or abnormal? It's normal. Fine, it's normally pale yellow. Now, if it becomes deep yellow, then definitely body has been dehydrated. So that problem can be solved by having a good amount of water intake. Can help that problem resolve. But if there has been some black colored urine, that's a problem. And what is the condition if the urine turns black? What is the condition when the urine turns black? The condition is called alkaptonuria. Alkaptonuria. The urine turns black, and that is an abnormality. So inside urine, there is a component called alkapton. Alkapton. Fine? We can replace this K with C also, alkapton. And that is a metabolic disorder. And due to that, the urine can turn black. So what is the normal color of the urine? Yellow, pale yellow is a normal color. And that is all due to what? A normal process called hemolysis. But for that, how it is going to take place every after 120 days, because after 120 days, the RBC which has been formed, they are going to die. Who is going to name the organ which is responsible for hemolysis? It's liver. But after death, what is going to happen? After death of this RBC, this dead RBC is going to be reserved where? Inside spleen. Is going to be reserved inside spleen. So spleen is called as graveyard of RBC. Spleen is called as graveyard of RBC. Graveyard of RBC. Is this clear? Fine. So that is it. Sometimes what happens? The spleen gets inflated. We call it splenomegaly. We call it splenomegaly. Fine. Sometimes the liver gets inflated. We call the term as hepatomegaly. Hepatomegaly. Megaly means inflammation. Megaly means inflammation. So inflammation of an organ. It's called megaly, inflammation. And this too occurs if the person is having liver cirrhosis. Fine? I think you have heard about this term. Liver cirrhosis in class 12. You have heard about this term about alcohol drinking, alcohol usage can lead into liver cirrhosis. But there is another one called non-alcoholic liver cirrhosis. There's another one, it's normally occurs due to alcohol consumption, called alcoholic liver cirrhosis, and sometimes it is non-alcoholic liver cirrhosis. Non-alcoholic liver cirrhosis. In liver cirrhosis, normally spleen gets enlarged in size and liver gets enlarged in size. Fine. The different types of anemia. One of the anemia is hemoplastic anemia. So people who are suffering from autoimmune disorder like Crohn's, they can have hemoplastic anemia where the RBC is going to die. Fine, before they become mature. So what is going to happen to the body? The body is not going to have enough amount of, the organs are not going to get enough amount of oxygen. So the body is going to always remain what? Lethargic, inactive, because organs are not going to perform to their highest threshold. Okay, so that is all about RBC. I think we have talked about RBC. I don't think we have missed anything. Yes, RBC. Anything that we have missed? I don't think we have missed anything about RBC. Fine. Yes. Let's get into the next one. What is that? And that is WBC. That is WBC. What is the other term for WBC? It's called what? It's called white blood corpuscles, or also called as leukocytes. The term cytes means cell. It has come from the term called cytology. And leuco means what? Leuco means colorless. Leuco means white. Leuco means white. So white colored corpuscles, white blood corpuscles. Again, when we are talking about calling them as corpuscles, that means they are not produced inside. They never show cell division. So they are produced inside bone marrow. They are produced inside bone marrow. Bone marrow. Okay, what is the origin? The origin is white bone marrow. The origin is white bone marrow. Which part of the bone marrow is white? So if this is the bone, these two parts are red bone marrow. So white bone marrow is present normally in this two lateral side, of the bone. Fine. So this is white bone marrow. What is the first cell? The first cell is called initial cell, or also called as stem cell. And what is the stem cell for WBC? The stem cell for WBC is called myeloblast cell. The stem cell for WBC is called myeloblast cell. Now, when we talk about WBC, they act as what? They act as soldiers of the body. They act as soldiers of body. That means they help in protection. Their main function is protection in the body. The main function is protection in the body. Yes, I was just inquiring about whether we have missed anything in RBC or not. One thing that we have missed is RBC, as because they have got hemoglobin inside. Say this is hemoglobin. This part is heme called iron, and say this is a long chain called globin protein. And globin protein is made up of 2 alpha and 2 beta chain. 2 alpha and 2 beta chain. And you know where each alpha chain contains 141 amino acid. Each beta chain contains 146 amino acid. So the molecular weight of this globin protein is huge. That is, hemoglobin is huge. And that is close to 59,000 Dalton. Approximately, they have got higher molecular weight in the form of 59,000 Dalton. What kind of protein that we have studied about hemoglobin in biomolecular topic? It's a type of quaternary protein. The highest level of complexity is present in hemoglobin. Fine. Now, this hemoglobin, they have got, so globin protein is made up of amino acid. So there are many of the amino acid. One of the amino acid is histidine. And this histidine is having negatively charged. Now, it is going to always bind with what? H plus. Now, where from H plus is going to come? Inside RBC. Inside RBC. Inside RBC, if this is the RBC and these are the tissues, these are your thigh muscles tissues, and say you are breaking down glucose and you are running at a great speed. Clear? Now, you are going to break down this glucose, will be broken down in terms of oxygen to give rise to 6 molecules of carbon dioxide plus 6 molecules of water plus 686 kilocalorie of energy is going to come out, which is equal to 38 ATP. Just now we have talked about this. Now, this carbon dioxide will be pushed inside the RBC. So the moment the carbon dioxide comes inside RBC, the carbon dioxide binds with water. Tell me the name of the enzyme. Carbonic anhydrase, which is the fastest amino acid. This carbonic anhydrase binds with this carbonic anhydrase helps the carbon dioxide bind with water and makes H2CO3 carbonic acid. And this carbonic acid breaks down into H plus HCO3 minus plus HCO3 minus. This bicarbonates, this is the bicarbonates where 70-75% of the carbon dioxide is transported in the form of bicarbonate ion. That means more the carbon dioxide transport, more is the formation of H plus ion. And this H plus ion is going to make the whole RBC or the blood, what? More of H plus is going to make it acidic. So the blood is going to become acidic. And if the blood becomes acidic, the condition will be called as acidosis. And that what really occurs in septicemia, when your body is completely infected by huge amount of bacteria, your body turns into septicemia where the blood becomes acidic. So your blood having more of H plus ion. So this H plus ion has to be arrested. Who is going to arrest this H plus ion? Inside RBC, this is a hemoglobin, and in hemoglobin, there is this amino acid called histidine, and this histidine is having heavy charge. It is going to arrest on this H plus ion and make the blood neutral. So it tries to maintain acid-base balance. One of the bestest function of blood, especially RBC, is to act as buffering agent. So it helps in acid-base balance. So one of the best function of RBC is to act as buffering. They act as a buffer. That means they try to regulate the acid-base balance. Got the point? So this is about RBC. This was left behind. Okay, let's come back to WBC. So soldiers of the body. So WBC are what? Soldiers of the body. So in Bengal, we can call WBC as West Bengal Corps. West Bengal Corps. So they are soldiers of the body. Now, so when we talk about WBC are of two types. WBC are of two types. They are, the first one, so star point. Why? Because you can expect questions from this area. So WBC are of two types. One is called granulocyte. One is called granulocytes, and the one is called agranulocytes. Agranulocytes. So what is granulocytes? Granules present inside cells. Granules present inside cells are granulocytes. That means they are also called as, see, they are also called as PMNL. What is PMNL? They are called as polymorphonuclear leukocytes. They are called as polymorphonuclear leukocytes. Polymorphonuclear leukocytes. Now, what is the meaning of poly? Poly means many. Morph means form. Nuclear means you know that nucleus, and they are having different forms of nucleus. We can classify them into three types. And what are they? I will talk about that. A means without granules inside cells. So they are without granules. So there are two different types. What are they? Never eat banana. Never eat banana like monkeys. These are the 5 different types of WBC. These are the 5 different types of WBC, where N stands for neutrophil. N stands for neutrophils. E for eosinophils. B for basophils. B for basophils. Okay, like for lymphocytes, and monkey for monocytes. Monkey for monocytes. So these are the 5 different types of what? 5 different types of WBC. Okay, now 5 different WBC. What about polymorphonuclear leukocytes? When we talk about eosinophils, they are also called as acidophils. 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 Okay. Now, why they are called acidophils? Phil means loving, like hydrophilic, hydrophobic. Phobic means hating. Phil means loving. So when we talk about acidophil, that means they are called as acid-loving cells. These acid-loving cells are also called as eosinophils. Eosin-loving cells also. So eosin-loving cells, that means these acidophils are also called as eosinophils. Why they are called eosinophils? Because they can be stained by an acidic stain named eosin. So what is the stain? The stain is acidic stain, and that is eosin. So you can see over here, acid loving, basic loving basophils, neutral loving neutrophils. So acid, base, neutral. Clear? So they can be stained by an acidic stain called eosin. And what is the function of these eosinophils? They can make allergical reactions in your body. They can make allergical reactions inside your body. They can make allergical reactions inside your body. Okay? Allergical reactions inside your body. And do you think this allergy is good? Yes, it is good. How it is good? Because the function of WBC is to help in protection. So their main function is what? Their main function is to protect the body. And how they can protect the body is by making what? Giving you some idea. They are going to make some rashes in your body the moment if you eat on some of you are allergic towards brinjal, some of you are allergic towards eggs. Fine. So the moment you take that egg and if you're allergic towards that, your eosinophil is going to help you understand that you should not take this particular food again. So that is the way, because what is the function? What is the definition of allergy? Allergy means hypersensitiveness to something. Sometimes you get allergic seeing one of your friends. So that is also what? Allergy. So think about it. So this is called allergical reaction. Now, that means you have got a good amount of eosinophils in your body if you are allergic towards somebody. Any issues? Good. Now, here, there is a condition called eosinophilia. When you become doctors, you are going to understand that. Eosinophilia. What is going to happen over there? In eosinophilia, this is a term. Just find it out. Check it out. What is the meaning of the term eosinophilia? It's a medical term. Okay. Now, when we talk about basophils, that means they can be stained by a basic stain. And what is the name of that basic stain? That is methylene. Methylene. So methylene blue is a stain, and this methylene blue is a basic stain. So they can be stained by a basic stain named methylene blue. What is the function of basophils? The function of basophils is they help in H square. This is how you can remember it. H square. So they give rise to, they can form two things. One is histamine and heparin. Histamine and heparin. What is histamine? Histamine acts as vasodilator. They act as vasodilator. What is the meaning of vaso? Vaso means blood vessel. So they can help in increasing the diameter of the blood vessel. They can help in increasing the diameter of the blood vessel. If the diameter of the blood vessel increases, and when this is really needed, that means this is the real cause of what? Inflammation. This is the real cause of inflammation. Yes, I think up till this age you have experienced slap from somebody. I think it's better to get slapped by your parents rather than your friends. Okay. Now, if you get it and you are not ready, because if result is declared and you get it, then your body is prepared for that. Fine. But suddenly if you get hit, what happens with this part of the face where you got slapped? This part of the face is going to get inflated. Inflammation is going to occur. That is what has happened inside the blood vessels. The blood vessels has felt something has went wrong. So suddenly if you get hit somewhere, your body gets inflated because the body understands that something is going wrong. And that part of the body is highly vulnerable towards infection. So what is going to happen? If the soldiers needs to reach over there in that part, soldiers in WBC needs to reach to that part, It can reach easily if the road has been made dilated. What is the road? The blood vessel. Got the point? So this inflammation is good or bad? It's good. Got the point? So whatever happens in your body automatically is for to save your body. So basophils, basically they secrete histamine, and this histamine is also secreted by a corrective cell. What is that? I'm going to study that. Mast cells. Come on, it's there in your book. Mast cell. Mast cell, they can also secrete histamine, and they can help in vasodilation. They can help in vasodilation. And there is a perfect statement that mast cell is must for inflammation. Mast cell is must for inflammation. It's a perfect statement. Without mast cell, you cannot have inflammation. Is inflammation is good? Not always, because majority of us, we are suffering from inflammatory diseases like osteoarthritis, which is an osteoarthritis, rheumatoid arthritis. These are all not osteo, but rheumatoid arthritis is an example of autoimmune disease that you have studied in class 12. Fine. What is autoimmunity? When your cells, fails to recognize the normal cells, and they start considering them to be an antigen and fights against those and kills them, kills the healthy cells. So that is called autoimmunity. So in autoimmunity also, what is the problem? The problem is inflammation. In that case, inflammation is not good. Clear? Achha. When you feed on turmeric during COVID time, what was the normal food that we used to have? Always, we have said this particular food contains anti-inflammatory, and they can increase the immunity of the body. So the best example of that was what? Turmeric. Turmeric is anti-inflammatory. Got the point? So right now, whatever food if you are having healthy foods, that means you are taking anti-inflammatory foods basically. Clear? And if the inflammation occurs, what is going to happen to the blood vessels? The blood vessel gets dilated, and just among the blood vessels, what are present? The nerves are also present over there. So the nerves are present at the top of the blood. So that is what is inflammation. So histamine is good sometimes. Why? Why sometimes it's good? Because see, the blood vessels, say inside the blood vessels, there has been some deposition of cholesterol or calcium. Cholesterol or calcium. Tell me, what is the condition called? If the inside the blood vessels, the cholesterol gets deposited, what is the condition called? Together we call it CAD. Together we call it CAD. What is CAD? CAD is called coronary arterial disease. Coronary arterial disease. And there are two different types. One is called arteriosclerosis. One is called arteriosclerosis, and other one is called atherosclerosis. Atherosclerosis. When we talk about sclerosis, that means we are talking about plaque. We are talking about deposition. So arteriosclerosis means arteriosclerosis means what? Calcium deposition. Calcium deposition. Now tell me, Shojit, what is the common between calcium and arterio? Both of them are 7 letters word. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. So if calcium deposition occurs, we call it arteriosclerosis. If cholesterol deposition occurs, we call it atherosclerosis. If cholesterol deposition occurs, we call it atherosclerosis. Any issues? Clear? Now, so this is a cholesterol or calcium present over here. But if there is sudden dilation, what is going to happen? See, the normal chances is the blood is not going to be able to pass through due to blockage. But you know what? When you become doctor, the moment you get this kind of patient, what is the first injection you are going to give them? Come on. In class 12, we have studied that. In the chapter called Microbes in Human Welfare. Statins. You are going to initially put statins or clot buster. Clot has already occurred. Clot buster called streptokinase. What is the bacteria where from you can get that? Streptococcus. No. streptokinase, you're going to get it from Streptococcus bacteria. And statins you're going to get. Statins are what? Blood cholesterol lowering agent. Remember that? Blood cholesterol lowering agent. And it is produced from Monascus purpureus. It's a yeast. It's a fungus called yeast. Unicellular fungus called yeast. And what is the agent? Statin. What is the agent? Statin. So it's a blood cholesterol lowering agent. Now, if this happens, the moment you inject histamine, what is going to happen immediately to the patient? The blood vessel is going to get dilated, and this particular deposition is going to come over here, and there will be huge, you are providing space for the blood to reach towards the heart. Got the point? So the person, you can save the person from heart attack. Clear? When you become doctors, you are going to study, you are going to give a patient, if you feel a patient is suffering from cardiac arrest, you are going to give the injection called GTN, and it's a complete vasodilation. So you are going to keep with the saline, you are going to put that injection so that the blood vessels remains dilated. So if the blood vessels remains dilated, you are not being able to take away the particular reason of the clot. It can be either cholesterol deposition or calcium deposition. So you're not treating that. You're not being able to treat that because you have still not performed angiography. First, you have to go for angiography to check what is the site where the deposition has occurred. To check that, you have to go for angiography first. And then when you find that there are two areas where angioplasty needs to be done, balloon angioplasty needs to be done, then you're going to do that. And if the blockage has been in many of the areas, you're not going to go for the angioplasty. So what is the take now? Bypass surgery. The only thing left is now go for bypass surgery. Okay, we'll talk about all those in circulatory system, but histamine. Got the idea about histamine? It's a vasodilator produced from vessels. What is heparin? Heparin are anticoagulants. Anticoagulants. That means they are never going to allow the blood to clot inside the blood vessels. They are not going to allow the blood to clot inside the blood vessels. Now tell me, what are the reasons? Do you really want blood clot inside your blood vessels? So what is the answer to the question? Why blood never clots inside the blood vessels? Inside the blood vessels. Tell me, what is the reason for that? There are four main reasons. For that. The first is there is a continuous blood flow. There is a continuous blood flow inside the blood vessels. So in any point of time, the blood vessels have blood, and there is a continuous pumping occurring from by the help of the heart. This is never going to allow the blood vessels without blood. So there is a continuous flow of blood. So the RBC, WBC, platelet, they don't have any, they don't get to stuck. So they are always in a moving condition. So there is no chances of clot inside the blood vessels. So there is a continuous blood flow. Second, there is a presence of anticoagulant inside blood vessels. What is that? Heparin. So we are now talking about heparin, which acts as an anticoagulant. They are going to never allow the blood to clot inside the blood vessels. Now, what is the position? What is the location of this blood vessel, this anticoagulant? If this is blood vessel, the anticoagulant heparin are present as they are mucopolysaccharides, heparin, and the flowing blood, when it comes in contact with this heparin, they are never going to allow the blood to clot. But the moment there has been hemorrhage, the blood vessel has been burst and the blood has come out from the blood vessel. Over here, there is no presence of heparin. So they are going to clot out immediately when it comes out. And what is the term I have used for the bursting of the blood vessels? Hemorrhage. What is the term? I think you have heard about the term called internal hemorrhage inside brain. Come on. Hemorrhage. Hemorrhage. Okay. Rage means bursting of the blood vessels. Clear? Now, So the second point is presence of anticoagulant called heparin. The third point is presence of the inner lining of the squamous epithelium is very thin and smooth. So due to the smoothness of the endothelium, I think we have talked about in this chapter only what is called endothelium. Endothelium of the blood vessels. They are lined by squamous epithelium. They are lined by squamous epithelium, and squamous epithelium, you know that one of the features of squamous epithelium, they are very thin, broad and flat cells. Along with being flat, they have got what? They are very smooth. They are very smooth, just like the inner lining of your cheek cells, the inner lining of your mouth. They are also having squamous epithelium. So what makes the blood vessels so very smooth? The squamous lining. So RBC, even if they want to stay over here, they are not going to be able to stay. Why? Because it's very slippery. So thin and smooth endothelium lining never allows the RBC to make Rowley formation. We have talked about sometimes RBC gets stuck. Sometimes RBC remains like stacks. Is this clear? So that is about the third point. And the fourth point is what? They have got some large anions. They have got some large anions in the form of Cl- and HCO3-, which inhibits the action of what? Coagulating factors. Action of coagulating factors. So we are going to study about 13 coagulating factors. Fine? Okay. So these large anions, they can inhibit the function of coagulating factors. These are the reasons of why blood never clots inside the blood vessels. If any one of them is showing problem, then definitely the blood vessels can have, there's every chance that the blood vessels can have what? Coagulation inside the blood vessels. What is the term called? If that happens inside the coronary artery, what is that blood clot inside the blood vessels called? What is the medical term for that? Hematoma is a local clot. What is the term where the blood gets clot inside blood vessels? Thrombus. I think you've heard about coronary thrombosis. You've heard about coronary thrombosis. What is thrombosis? It has come from what? What is thrombus? Is a blood clot inside the blood vessel. And if this occurs inside the coronary artery, which supplies the blood towards the heart muscles, if this occurs inside the coronary artery, then the person will be called to be suffering from coronary thrombosis, and that will lead into what? Heart attack. Clear? Okay. So let's come back. So we are talking about basophils right now. So basophils is over. So it helps in anticoagulation. So it never allows the blood to clot. What is the function of neutrophil? The function of neutrophils, that is they neutral loving cells. That means they can be stained both by acidic as well as basic stain. Now, neutrophils basically they help in phagocytosis. They can feed on the foreign antigens. Is this clear? So the process by which they can feed on is called phagocytosis. Any issues with this? Now, what is the function of lymphocytes? So basically, why they are called polymorphonuclear leukocytes? Let us explain that. Neutrophils, they have got multi-lobated nucleus. Many lobes inside the nucleus. Eosinophils, they are having bilobed nucleus. Clear? This is the nucleus, bilobed nucleus. They have bilobed nucleus. What about basophils? They have got trilobed nucleus. They look like S-sepid. This is one lobe. This is another lobe. And over here there is another lobe. So they are trilobed nucleus. Clear? Trilobed nucleus or S-sepid nucleus is there in the basophil cells. So that's why they are called polymorphonuclear leukocytes. This is very, very important as far as Neet is concerned. Okay. Now come down to organocytes. So they don't have granules. They are lymphocytes and monocytes. When we talk about monocytes, they are also called as macrophage. They are also called as macrophage. What is the meaning of the term macro? Macro means large sized. But you know what? These neutrophils are called microphage. They are called microphage. They are called microphage. So they are smaller sized and they are wandering cells. Their number are huge. Neutrophils in blood, they are when we talk about WBC, they are close to 55 to 65% of the blood is neutrophils. So they are also called as wandering cells. We will talk about that. Macrophage are the largest WBC. Macrophage are called as the largest WBC and they can kill the foreign bodies. When we talk about lymphocytes, they have got huge importance. Why? Because they can help in the formation of antibodies. They can help in the formation of antibodies. And in class 12, we have studied about Y-shaped antibody. I think you have done it. Heavy chain and light chains. Though we are going to talk about that in lymphocytes, we are going to go in depth of it. So they help in antibody formation and also they can increase immunity of the body. They can increase the immunity of the body. So they help in increasing the immunity of the body. Is this clear? Okay, done. Now, when we talk about monocytes, again, let me tell you, they are horseshoe-shaped. They have got horseshoe-shaped nucleus. Horseshoe-shaped nucleus. It's very important about monocytes. Any issues about WBC? I think this part is over. Now, when we talk about the numbers, of WBC, WBC number is close to 6,000 to 8,000 per cubic millimeter of blood. So if you compare between RBC and WBC, they were in lakhs. RBC were in lakhs. Now think about WBC, they are now in thousands. So when we talk about the ratio between RBC is to WBC, their count is going to come close towards 700:1 approximately. 700:1. Now, in case of male and female, the WBC count is all the same. Now, if your body is having huge number of soldiers suddenly, that means your body is having 12,000 to 15,000 of WBCs. That means in your body, the soldiers count has been increased. Now, what is the need for the increased soldier count? Definitely your body is infected. Your body is having some antigens, and to combat with those, the number needs to be increased. So definitely, if the number of WBC has been increased, the condition will be called as leukocytosis. If the number reduces than normal, the condition will be called as leukopenia. It was in case of RBC, this condition was erythrocytopenia. In case of RBC, the increase in RBC count was called as erythrocytosis. Now, if the number increases even more than 70,000 to 80,000 and even 1 lakh, per cubic meter of blood, the condition will be called as leukemia. Emia means blood. Leukemia is nothing but blood cancer. It is also called as blood cancer. Fine. Now, in WBC, the most important point is about this. And the term is called diapedesis. What happens in diapedesis? Say, for example, this is the skin. The first line of defense is skin for your body. Just like the border city force of the nation is the first line of defense. And say this part of the skin has been infected by bacteria. Now, this is the blood vessel. And inside the blood vessel there are squamous epithelium. And these blood vessels is like this. Now see, if this is WBC, they are nucleated. WBC can squeeze themselves in between the two epithelial cells, just like amoeba. They can squeeze themselves out from the blood vessels to the infected area, so they can be straight away to the infected area and fight with those bacteria. It's good for your body or bad for your body? It's always good for your body. So if it would have taken the long way to reach, it would have taken much longer time to reach over there. So it can squeeze themselves out in the form of amoeboid movement, So they can come out of the blood vessels in the form of amoeboid movement and reach towards the infected area. The process by which they can do this is called diapedesis. So diapedesis is only shown by WBC. It's a very, very unique feature of WBC. So what is that? The process by which WBC can come out of the blood vessels to the infected area by amoeboid movement, Is this clear? Very good. Now let me get into the thrombocytes. Fine. Let me send you this first. Okay. RBC, WBC, YT. Okay. Now, let us start with thrombocytes. Thrombocytes, otherwise called as platelets. First, tell me a disease where the platelets comes down drastically. Chikungunya and dengue. Okay, the platelet count reduces drastically. Tell me, the thrombocytes which are called platelets, what is the initial cell? What is the origin? The origin is bone marrow. Fine. Next point. What is the initial cell? The initial cell or stem cell is called megakaryocyte. Cells. Okay. Next point. What is the function? Their main function is to help in blood coagulation. What is the number? The number in case of male and female is all the same. It is 1.5 to 2.5 lakhs per cubic millimeter of blood. Go by NCERT, whatever this is about this. 1.5 to 2.5 lakhs per cubic meter of blood. If the number increases the normal, the condition will be called as thrombocytosis. If the number comes down, we call it thrombocytopenia. Penia, when the number comes down, is called penia. Now, the process of blood coagulation. Inside our blood, what is present? Calcium ion as the cations. Calcium ion is present inside our blood. This calcium ion binds with the blood protein called prothrombin. When we talk about prothrombin, pro means inactive. So they remains in the form of inactive. Now, the moment there has been a hemorrhage in the blood vessels, this enzyme gets activated, named thrombokinase gets activated in presence of magnesium ion. This thrombokinase is going to help the prothrombin turn into active thrombin. And this active thrombin now binds with the inactive blood protein present already inside the plasma called fibrinogen, and this fibrinogen is inactive. is inactive. And this fibrinogen, when binds with thrombin, it becomes active fibrin. It becomes active fibrin. Active fibrin. And what is fibrin? These are fiber-like structures. These are fiber-like structures. And within this meshwork of fibers, if the RBC gets clogged, see, the RBC gets clogged, RBC gets stuck. The moment RBC gets stuck, what is going to happen? That is, active fibrin plus RBC makes blood clot, And for this blood coagulation, the time requirement is 9 to 11 minutes. This is the blood coagulation time. This is called as blood coagulation. Blood coagulation time is 9 to 11 minutes. And let me tell you, there is a term called bleeding time. There is a term called bleeding time. And that is 4 to 6 minutes. It all depends upon the type of bleeding, the type of cut, if there has been. So 4 to 6 minutes is a normal bleeding time, but the normal coagulation time is 9 to 11 minutes. And there are some blood coagulating factors. But before we get into the blood coagulation factor, how the anticoagulants are going to work, let us find it out. How the anticoagulants work. Normally we know that there are two types of anticoagulants. One is natural anticoagulants. One is natural and the one is artificial. Natural and another one is artificial. So when we talk about natural one, the natural example is the first one, heparin, and the second one, hirudin. The hirudin are present where? Name the organism where the anticoagulant hirudin is present. It is hirudinaria. There's leech. There's leech. So inside leech they have got hirudin, and from that term only we call them as hirudinaria granulosa, the scientific name of leech. The scientific name of leech. Now what is heparin? Heparin are the natural anticoagulants. And how they work? Let us find it out. How they work. The heparin never allows the prothrombin to become active thrombin. This is where the heparin works. Heparin never allows the prothrombin to become active thrombin. And that is why the heparin inside the blood vessels, they never help in blood coagulation. Got the point? So if there is no thrombin, the process is not going to proceed. This is how they stop the blood coagulation. How the artificial anticoagulants work, and what are those artificial anticoagulants? Number one, the artificial anticoagulant number one is sodium citrate. Sodium citrate and sodium oxalate. Sodium citrate and sodium oxalate are the artificial anticoagulants. Sodium citrate takes off the calcium from the system and makes calcium citrate plus sodium. The calcium never remains in an ionic form. They get precipitated inside the blood. If there is no calcium ion present, do you think the process will start? The process never starts. So this is how the blood coagulation is prevented. This is how the blood coagulation is prevented, by the sodium citrate and sodium oxalate. And what is the purpose of this artificial anticoagulant? During blood transfusion, when the blood is being taken out, when you donate blood, inside that pouch already, the powder called sodium citrate is being administered. So that sodium citrate is not going to help the blood to clot, because within 9 to 11 minutes, the blood is automatically going to get clot when it comes out of your blood vessels. Got the point? Now, secondly, when a person comes down to your home to take the blood for any type of diagnostic test, so inside that pouch, inside that vial, what is being kept priorly? The powder of sodium citrate or sodium oxalate. The moment the blood is being given over there, they take that blood inside that vial. That calcium of the blood is never going to get in a calcium ionic stage. They get precipitated into calcium citrate or calcium oxalate. Got the point? There's another one which is called EDTA. What is EDTA? It's another type of anticoagulant. The name of that anticoagulant is ethylene diamine, tetraacetic acid, tetraacetic acid. This is EDTA. How it works? It binds with calcium and makes chelate formation. I think you know what is called chelation. It arrests the calcium, just like in alum. Fine, in alum the same thing, chelation occurs. Got the point? So this is how they work. Now let's come into the next stage called the 13 coagulating factors. And you have to remember it. There's no other way out. And see the sequence also you have to remember. Fine. The first one is prothrombin. And prothrombin is already there in the blood coagulation step. Prothrombin is the first one. This is the Roman one, the first sequence. The second one is fibrinogen. Fibrinogen is also there in the blood coagulation. The second factor is fibrinogen. The third factor is thromboplastin, thrombokinase. Thromboplastin or thrombokinase enzyme thrombokinase enzyme. The fourth one is calcium ion. The fifth one is proaxillarine. The sixth one is called axillarine. The seventh one is called SPCA, serum prothrombin converter accelerator, SPCA. They are also called as stable factor. This is 7. 8 is anti-hemophilic factor A. 8th, the ninth one is anti-hemophilic factor B, which is also called as Christmas factor. The 10th one is called Stewart power factor. 11th is plasma thromboplastin antecedent called PTA. The 12th one is Hageman factor. 13th is about fibrin stabilizing factor. Fibrin stabilizing factor, or also called as FSF. This is what you have to remember. All the 13 coagulating factors. You have to remember it. There's no other way out. Clear? Yes. So just write down once or twice. You are going to have it. Fine. So these are the different types of blood coagulation factor which normally helps in blood coagulation. What is the disease where the platelet count decreases suddenly? It is due to dengue. And dengue is caused by the, what is the vector? A female Aedes mosquito, female Aedes aegypti. Both chikungunya and dengue. And tell me, It is caused by a virus. What is the name of the virus? What is the group of virus they come under? Flavivirus. Flavivirus. Okay. So that's all about what? About the blood coloring factors, and also we have been able to cover the whole of the blood part. So blood is a fluid connective tissue. So next day what we will do is about bone. About bone. But before that, let us solve some of the questions. Let us solve some of the questions. This is about platelets. Platelets. Let us solve some of the questions. Okay. Okay. Let us solve some of the questions. Let us find out. Give me your time. Yes. The moment you stop this. Okay. Quick revise of some of the questions. Let us do that. Yes. Okay, fine. Go for this. What is written? Mast cells. The first question. Mast cells of connective tissues contain vasopressin and relaxin, heparin and histamine, heparin and calcitonin, serotonin and melanin. Heparin and histamine. Very good. Heparin and histamine. Next, which one of the following contains the largest of extracellular material? Largest of extracellular material. Myelinated nerve fiber, striated muscle, alveolar tissue, stratified epithelium. Which one is connective tissue out of this? Largest extracellular material are present in alveolar tissue, and that's why they act as a packaging material. That's why they act as a packaging material. Okay. Which cartilage is present at the end of long bones? Have you talked about cartilage? End of long bones? Hyaline cartilage. Huh? Yes. During an injury, nasal septum gets damaged. At recovery, which cartilage is preferred? Nasal. Nasal contains what? Elastic cartilage. Nasal is having elastic cartilage. We won't talk about that. Fine. Cartilage we have not completed. Cartilage we have completed. So we can easily do it. Come on. Elastic. They have got what? Elastin. They have got what is the protein present? Yellow protein. Yellow fibers. Elastin protein. So elastic cartilage. Which cells do not form layer and remains structurally separated? Do not form layer and remain structurally separated. Do not form layer. Yes. Do not form layers. Very good. Nerve cells, they do not form layers. The kind of tissue that forms a supportive structure in our pinna. The kind of tissue that forms a supportive structure. Okay. The kind of tissue that forms the supportive structure in our pinna. External ear is also found in nails, tip of the nose, ear ossicles, vertebrae. The kind of tissue that forms the supportive structure in our pinna, external ear, is also found in tip of the nose, because what is common between elastic cartilage? What is common? The common is elastic cartilage. Which one of the following is correct pairing of part and the kind of muscle tissue that moves it? Which one of the following is correct pairing of part and the kind of muscle tissue that moves it? Pairing of part and the kind of muscle tissue that moves it. Biceps of upper arm, smooth. Never. Okay. Abdominal wall, smooth muscles. Yes. Iris, involuntary smooth muscles. Iris, yes. Heart wall, involuntary unstriated or striated. So the answer is between the abdominal wall, smooth muscles, and iris, involuntary smooth muscles. What is going to be? Involuntary smooth muscles. Iris. Iris is dilated. Iris or pupil? Which one gets dilated? So what is the answer going to be? Abdominal wall. Okay. In which of the following preparations are you to come across cell junctions? Cell junctions frequently. Which of the following preparations are you likely to come across cell junctions most frequently? Cell junctions. Cell junctions. Junctions, that means we are talking about what? Connective. Connective? No. Tight junctions, gap junctions are present where? So what is the answer going to be? Tendon is what? Tendon, hyaline cartilage, thrombocytes. Thrombocytes ruled out. Ciliated epithelium is an epithelial tissue. And think about the junctions you have talked about is about what? The junctions that you have talked about is under epithelial tissues. Epithelial tissues. So the answer is going to be areolar connective tissue joints. Areolar connective tissue joints. Huh? Areolar? Come on, how to remember areolar tissues? Sam. A for areolar. A for areolar. S for skin. And M for muscles. So it connects skin with muscles. So is there any skins with muscles over here? So the answer is integument. That is skin with muscle. Sam. So always remember areolar tissue by Sam. Next. Yes, structure separated. Mast cells. Come on. This one. The polysaccharide present in the matrix of cartilage is known as matrix of cartilage. Chondroitin or casein? Casein is there in the milk. So answer is chondroitin. Chondroitin. Fine. The answer is chondroitin. Tell me simple epithelium. And you know that. Simple epithelium. In which the cells are hardened. Is a tissue in which the cells are hardened and provide support to the organs. Cemented directly to one another to form a single layer. Continuously dividing and help in organ function as organ. Loosely connected. B is the cemented directly to one another to form a single layer. epithelial tissue with thin flat cells appearing like packed tiles. Packed tiles, pavement. Kire, pavement. We have talked about pavement epithelium. And you know what? Squamous epithelium looks like pavement epithelium because they bind together like tiles. So answer is inner cells of inner lining of cheek. Protein present. Chondrine. Protein present inside the matrix of a cartilage is called chondrine. In case of bone, it is going to be ossein. Basement membrane is made up to. Basement membrane is made up of both epidermal and endodermal. And basal lamina. No cell at all, but is a product of the cells. No cell at all. Basement membrane is no cell. It's acellular. Basement membrane is acellular. It's a secretion of epithelial tissue as well as connective tissue. So answer is going to be D. Clear? Stratum germinativum. Germinativum is an example of which kind of epithelium? Cuboidal, columnar, ciliated, squamous. Stratum germinativum is an example of which kind of epithelium? Cuboidal, ciliated, columnar, and squamous. Answer is very close calls. It's a close call. Have a look in your book. They have given some structure of stratified epithelium. Stratified. You can see that. Compound epithelium. What is the structure they have given? Squamous or cuboidal? Look over here. What is the part of the skin? The part where there is a maximum wear and tear. Squamous. So squamous. Clear. Very good. But you have to find the best answer out of this, no? So, what is this? Okay. Histamine secreting cells are found in connective tissue, lungs, muscular tissue, nervous tissue, connective tissue. Formation of cartilage, bones involves. Go for it. Deposition of bony matter by osteoclast and resorption of by cardiochondroblast. Deposition of bony matter by osteoclast and resorption of chondroblast. Deposition of bony matter by osteoclast only. Deposition of bony matter by osteoblast and resorption of. Very good. Answer is A. Hair present in the skin are dead cells. Epidermal in origin are made up of living cells. Dermal in origin are made up of living cells. Dermal in origin and made up of dead cells. Hairs are always epidermis. Epidermal. So which one? First or second? Dead. Here, dead. Only the bulb is living. Tell me the last one. Shojit, tell me. Actively dividing cells of skin. Stratum, Malpighi or stratum? C is the answer. Clear? So next day we are going to start with the bones. Cartilage is over. We need to start from bones. So next day we are going to start with the bones. Fine? Sure. And we are going to solve some questions also. Sure. That's all for today.